Okay, so when I say mRNA, what's the first thing that pops into your head? For pretty much everyone, it's the vaccine, right? It's the tech that basically defined the pandemic. But what if I told you that its most famous job, getting our immune system all fired up, is the exact opposite of what it needs to do for its next great mission? Today, we're diving into the quiet, the stealthy, and the truly revolutionary future of mRNA.
Yeah. For most of us, mRNA wasn't even a word we knew, and then suddenly, bam, it was a global headline. The COVID-19 vaccines were an incredible success story, rolled out at a speed nobody thought was possible. They proved to the entire world that this technology was a seriously powerful tool for teaching our bodies how to fight back.
But here's the twist, and it's a big one. For a whole class of really devastating diseases, the goal of an mRNA therapy isn't to get the immune system's attention at all. The goal is the complete opposite. It needs to be a ghost. It has to sneak in, deliver its message, and then just vanish before the body's alarms can even think about going off.
To really get why this invisibility is so crucial, we've got to quickly revisit the story we all know, why the vaccine version has to be so loud. So let's do a quick recap. mRNA is our body's immune alarm.
The process, when you break it down, is pretty brilliant. In step 1, we get an injection of mRNA that's carrying the instructions for just a little piece of a virus. Then in step 2, our own cells get to work, reading those instructions and building that harmless protein. But the real magic happens at step 3. Our immune system spots this new protein and sounds the alarm, flagging it as an invader. That kicks off a full-scale response, creating the antibodies and T cells, our body's soldiers, which trains it for the real fight down the road.
And that's the key, right? For a vaccine, that immune reaction isn't some annoying side effect. It's the entire point. It's a feature, not a bug. You absolutely have to sound the alarm so your body's defenses can learn what the enemy looks like and be ready for a real invasion.
Right, so what happens when the enemy isn't an outside invader? What happens when the problem is actually a glitch inside our own system? Well, that's where the whole strategy completely flips on its head. We go from sounding the alarm to needing complete and total stealth.
Just think about genetic disorders for a second. In a lot of these conditions, the body's own DNA has a typo, a flaw, that stops it from making a protein it desperately needs. The problem isn't a virus, it's a missing part. So if you send in an mRNA message to build that missing protein, you've got a huge problem. The last thing you want is for the immune system to attack the very thing that's supposed to be the cure.
So this sets up two totally opposite missions for the exact same technology. You can see on the left, for vaccines, the mRNA is like a loud town crier, handing out a wanted poster for the immune system. But for therapeutics on the right, it has to be a secret agent, slipping past the guards, delivering a coded message, and then vanishing without a trace.
So how in the world do you make a molecule, one the body is literally designed to spot and destroy, invisible? Well, the answer came from a Nobel Prize-winning discovery that basically gave mRNA an invisibility cloak.
You know, this idea didn't just appear overnight. It's been cooking for a long, long time. mRNA was discovered way back in 1961, and by 1990, we were already using it to make proteins in mice. But for decades, that pesky immune response was the roadblock. The entire game changed in 2005. That's the breakthrough moment, when scientists figured out a tiny chemical tweak that let mRNA fly completely under the immune system's radar.
And this is the secret ingredient, pseudouridine. Look, think of RNA as being built from 4 different types of Lego bricks. Scientists discovered that if they just swapped out one of the normal bricks for this slightly modified version, pseudouridine, the whole molecule suddenly looked friendly to the body. It's the perfect disguise. The immune system's guards, which are on high alert for foreign RNA, just see it and let it pass right on by.
So armed with this molecular invisibility cloak, scientists could finally go after a whole new mission. Instead of fighting invaders from the outside, they could start fixing problems from within, specifically by replacing critical proteins that our bodies sometimes just can't make on their own.
And this is why it's so incredibly elegant. Instead of a patient needing constant injections of a missing protein, you just give them the mRNA instructions. Their own cells become these little on-demand factories, producing exactly what's needed, right where it's needed. It's a complete paradigm shift for treating genetic diseases.
And listen, this isn't science fiction. This is happening right now.
For these devastating metabolic disorders like OTCD and propionic acidemia, conditions where just one missing enzyme can lead to severe brain damage or worse, these stealth mRNA therapies are already in human clinical trials. We are right on the cusp of empowering the body to make the very thing it's missing.
But replacing proteins? That's just step one. The next frontier is even more ambitious. What if, instead of just delivering instructions for a replacement part, the mRNA delivered instructions for a tool? A molecular tool that could perform surgery directly on our DNA. And you've probably heard of this tool, CRISPR/Cas9. It's often called a pair of molecular scissors. Basically, it's a system you can program to find one specific spot in our genome and make a precise cut, which lets scientists disable a faulty gene or even replace it.
And this is where it all comes together. How do you get those molecular scissors inside the right cell? Well, stealth mRNA is the perfect delivery system. The mRNA delivers the instructions. The cell uses them to build the Cas9 scissors. The scissors do their job, cutting the DNA, and then the mRNA instructions just fade away. It's a clean, precise, hit-and-run mission. Let's look at a groundbreaking real-world example, a therapy called NTLA-2001. It targets this devastating disease where a faulty gene in the liver pumps out a toxic protein that damages the heart and nerves. The strategy is to permanently switch off that faulty gene. And to do it, it uses a tiny lipid bubble to deliver two key messages: the stealth mRNA that tells the cell how to make the CRISPR scissors, and a guide RNA that tells those scissors exactly where in our 3 billion-letter genome to make the cut. The results from the first human trial were, well, they were staggering. A single IV dose of this therapy resulted in an average reduction of that toxic protein by 87%. Let that sink in for a second. An 87% drop from a single treatment. This was the first time gene editing had ever been used like this inside the human body, and it was all made possible by the quiet power of stealth mRNA.
So, this is where we are. We started with mRNA as this loud alarm bell to train our immune system, but we've discovered its secret identity, a silent messenger that can turn our cells into pharmacies and deliver tools to literally edit our own genetic code. It's one technology with two opposite, but equally powerful faces, and it just makes you wonder, now that we can write these biological messages, what are we going to program ourselves to do next?